And you can hear me on this thing, yes? Somehow we're not at the beginning of this. Let me go back. Sorry. So thank you for the invitation. This is an algorithm-free talk. So those of you who, I'm a user of everything that all of you put together. I'm sort of downstream and not a very sophisticated user, so don't ask me hard questions. So I'm going to give you a lot of biology and a bit of what some of the challenges still are computationally in terms of understanding how small RNAs actually work in cells, and my studies are in bacteria. So we're interested in small RNAs in regulatory networks, and it's an appropriate special topic for this year, where we're celebrating 50 years after Charcot and Monod. We're trying to think about how you carry out regulation, and suggested that the repressor might in fact be RNA. It wasn't in that case, but now we know it can be. And one other Monod quote is that anything found to be true of E. coli must also be true of elephants. I'll be talking about E. coli, but the small RNAs that I'll be talking about are quite equivalent in terms of the kind of biology they do to microRNAs and RNAi in eukaryotes, although actually the computational challenges are rather different for the two of them, because these are processed down to small pieces and these aren't, so we have more space to look at. So in E. coli, there are still searches going on. There was one published, I think, this month in Genome Research, and so this may not be a full list, but I think it's an order of magnitude correct list in terms of how many non-coding RNAs we have, that is, RNAs that are encoded in the genome, don't encode proteins, and act in trans. So I'm not talking about cis-acting RNAs, things at the moment, on the order of about 2% of the open reading frames in E. coli. And the ones that I want to focus on today are the couple of dozen or so that bind a particular protein, HFQ, as a chaperone, and then act by pairing. So we know a lot about them if we know that they bind HFQ. We make the assumption if they bind this protein that they act by pairing and perturb the expression of a message. And so they're, again, the bacterial equivalent of microRNAs and RNAi. There are a lot we don't know very much about. If we get structures, maybe we would know more. But people are studying sort of on the one-by-one biology approaches. So again, just the final comparison between our E. coli RNAs and the eukaryotic microRNAs, and for every statement I make here, there's at least one exception that we know of in the bacterial world already, and there'll be more in both cases. The primary transcripts, for the most part, are about 100 nucleotides long, and they fold. This is a transcription terminator, so they're frequently made as a freestanding transcript with this as the terminator. They're not usually processed, although there are exceptions to that. The protein scaffold, so in the eukaryotic case, there's a longer RNA precursor that has to be processed in multiple steps. It has to be eventually loaded onto a protein scaffold. The same is true for us, except the protein scaffold that we know about is this single protein HFQ that I won't say too much about in this talk, unless somebody asks me about it. And then loaded on those scaffolds, there's pairing with targets. And the major difference for most of the bacterial cases is that we're pairing at the 5' end, usually in the UTR, but it can be also early in the open reading frame. And the seed, in terms of where it is on this RNA, is all over the place. So there are sometimes it's at the 5' end, sometimes as it's color-coded here, it's internally, and that makes it computationally harder to do some of the things that we'd like to be able to do. So there have been a lot of global searches. They started out as computational searches that look for conserved sequences in intergenic regions. I would say now probably the sort of deep sequencing approaches are powerful are the most powerful way to find these things. Obviously, there's a lot of computation associated with getting the data out of that. But let's assume we have our set of small RNAs that we found by global searches, and therefore we don't know much about them. So what I would like to be able to do is put those small RNAs into regulatory networks. And to do that, I have to know minimally what's upstream, that is, when it's being made, and downstream, who it's regulating. And we do both of those in my lab. We take a particular small RNA, somebody comes into the lab, I say, here's the list of things we don't know anything about, pick one. Do genetics or other, or sometimes look just at the sequence and find the upstream regulators, and we can do that genetically fairly straightforwardly. And then downstream, how are we going to find the targets? So there are computational approaches, but I would have to say they're far from optimal. They work on what we put in and work a little bit on other things. They give us maybe a few good suggestions, but a lot of false positives and a lot of things aren't found. Our standard experimental approach has been to overproduce the small RNA and then look on microarrays for changes in the message, because there are changes in the message, and I think I have put them here. So most of the outcomes, the standard outcome is negative regulation, sometimes by occluding the ribosome binding site, and degradation of this message, and I'll show you a couple of pieces of data that just show you that. In addition, we have positive regulation, and in the case of positive regulation, we're opening up the ribosome binding site and allowing the ribosome to come in, but again, the message is frequently more stable. So if we look on a microarray, we can see these changes, and that's the major way that we find targets. The targets also have to bind HFQ, so the small RNA binds HFQ, the target binds HFQ. The problem is that we don't know when we're done. Each of these small RNAs can have a few to many targets, and the more we look, the more targets we find. So I think there is not now a case of a single target for one of these small RNAs, and there are a number of examples where there are dozens of targets. And so if we want to really understand the regulatory network, how this outcome may affect the stress that first set this thing off, we'd really like to know everything that this thing is doing. And there are further complications, because I'll show you that a given target can have multiple small RNAs feeding into it. So my favorite, and one of our first examples, I'll just go through very quickly because we sort of understand it and it gives you the basics if you're not familiar with this field. So here's RIB. It was identified in a genome-wide search for conserved sequences in intergenic regions. Turns out to be a small RNA that binds HFQ. We know that it's transcriptionally regulated by a repressor protein that represses when iron is abundant in the cell and comes off the DNA and allows transcription when iron is depleted. The furregulin has been studied for many years and is involved in iron transport, bringing iron into the cell, and has this as well as part of it. I should move over. Okay. So is this microphone not working? This one. Okay. Is that better? Okay. So why am I wearing this? I won't. I'll take it off. Okay. So anyhow, sorry about that. I hope you could hear some of it back there. Okay. So here's the reversible RNA switch as it works in a cell. We chelate iron so the repressor comes off. The small RNA, these are northern blots, so we're measuring RNA. The small RNA is made very rapidly within a couple of minutes. If we return iron to the cell, then we stop making it, and it's destroyed very quickly. Here's one of its targets. This gives you some idea of the predicted pairing. We don't know how much of this is important. I would guess just this. So this stuff is probably irrelevant for regulation. And if you look at what happens to this message, it very rapidly disappears. It's destroyed. It's degraded by an RNase, and we know what that RNase is in E. coli. once the small RNA is no longer present, the message comes back. So this is all happening very quickly at the level of the messages. So when we did that microarray experiment, when Eric Nasi did it a number of years ago, we had a lot of targets. So we have 17 operons, some of them with multiple genes, and what they have in common is that they all encode iron-binding proteins. So this is one case where we sort of understand what the physiology is and what the small RNA is doing. These are central, it's turning off the ability of this organism to use a TCA cycle because there are a lot of iron-binding proteins here and it doesn't need this. There's less synthesis of these iron-binding proteins. Iron becomes available for essential proteins, and in fact there's a gene that's positively regulated and that brings iron into the cell. So we get this sort of circuit in this case that we think we understand, where when iron is limiting, we make the proteins that you need to scavenge iron, and you make the RNA that reduces iron load. So that's good. And as we've gone through these, sometimes we find upstream regulators, we find targets. We don't think we have every part of the target list, but they're in all kinds of stress pathways in major regulatory networks, and I'll tell you a little bit about a few of them and what our limits are. So, as I said, our computational approaches to finding the targets are still far from optimal. Our experimental approaches, where we pulse the small RNA by overexpressing it from a regulated promoter and monitor changes in messenger RNAs, it's been done on microarrays, it could be done by deep sequencing, means that we have to be able to see mRNA stability changes, and sometimes that isn't seen, and we need to have enough messenger RNA to see it change. So for genes that aren't well expressed, we don't see them well. So my lab over the last few years developed a sort of an inverse approach that I'm calling a sort of RNA landscapes. And it's based on the fact that we think we know all or close to all of the HFQ binding small RNAs, the pairing small RNAs in bacteria, so that we can use these as our starting point. And we're going to take the targets that we're interested in. So in this case, we're going to start with a target and say who regulates it at the small RNA level. And we choose our targets because they bind to HFQ or because they have conserved 5' UTRs. And then we make a translational reporter, and then we screen for the regulators. So the gene that I'm going to start out by telling you a little bit about is RpoS. It's a transcription factor, a sort of core transcription factor. What happened there? So this computer didn't like my symbol, the sigma symbol. So it's a sigma factor that binds to RNA polymerase and transcribes genes when the bacteria sort of gets into stress conditions or into stationary phase. It's regulated at every possible level. We study this level as well. And the first small RNA that we found in the lab, in fact, positively regulated it by pairing and opening up a hairpin. So there's an inhibitory hairpin that allows, that keeps translation low, and this small RNA and then that one, these were found sort of not by the global searches, did the same thing. They paired the same region, although they look rather different. And the region that, and these structures, I mean, this one is from protection studies, from probing in vitro. This is purely an M-fold. So if anybody wants to fold it better for me, I'd be happy to have a better structure. So those two were found generally, and now we're going to take all the small RNAs that we know about and look at them in a microtiter dish format. And Pierre Mandin did this, and what you get is this kind of landscape in which some small RNAs negatively regulate the output, and the two that we know of that positively regulate continue to positively regulate, and we found a new one. So we can very quickly screen this set of 25 to 30 small RNAs for their effect on a particular target, and we can perturb the expression of the target. And they all pair in the same region, but they all look different. So I told you a little bit about DSRA and RPRA, where different regions are pairing. This small RNA is one of the ones that's sort of is different from the rest in that it's processed down, it's cleaved, and the region that ends up pairing is at the 5' end. So I'm interested in the biology of the systems, why have multiple small RNA regulators all able to open up this hairpin, and the simple answer is that they do it under different conditions and that they also have other targets. So that's outlined in the next slide here. Just there's a circle for each of these small RNAs. They each have targets that are specific to them, that are negatively regulated. They're each regulated by different upstream transcriptional regulators. So this, we think, is involved in biofilm maturation, and this small RNA is expressed when cells settle on a surface. This one is made when cells are at low temperature. This one's made when they're at high oxygen, so it's repressed at low oxygen. And we don't always understand why the cell is doing this, but we can get a lot of combinatorial regulation this way. But it gets more complicated in terms of thinking about these networks. So this is a small RNA called MGRR that Kyung Moon in the lab studied and looked at the sequence and found a binding site for a two-component system. And what that two-component system, it's important in Salmonella and in E. coli. In Salmonella, it's absolutely needed for pathogenesis. And what it does is help to, one, modulate magnesium flux into the cell, and two, regulate the cell surface in such a way that it's resistant to antimicrobial peptides. So it helps the Salmonella resist the immune system and antimicrobial peptides when it infects. And now we have a small RNA as part of that. And that small RNA, so Kyung did her microarray study and found two targets in it, and I want to just talk a little bit about EptB. Here's the pairing again. In this case, it's relatively compact over the AUG and the ribosome binding site, and it's effective enough so that in a wild-type cell, we can't see that message at all. We can't measure it. But when we delete the small RNA, we can see it. So what does EptB do? It also modifies the cell surface. So we think this is important for in some way that we don't really understand in how the organism interacts with its host. So when Kyung did the RNA landscape experiment for that, she found the small RNA that we knew about, but we found another one, and this is the one that also regulates RpoS. So now we have, So now we have a lot of connections here. We have EptB is regulated by this guy. These are the upstream regulators for this small RNA. These are the upstream regulators for this one. They're also regulating EptB. And in work, I'm not going to tell you about. So all of these funny things should be sigmas. I don't know why your computer doesn't like sigmas, but so there's another specialized sigma factor in E. coli. It regulates yet another small RNA called MIC-A. MIC-A regulates these upstream regulators. So these things are all connected together in complicated ways, and in fact there are other connections. So those of you who are interested in modeling regulatory networks in bacteria like E. coli, where we think we know a lot, we're just beginning to understand all the additional connections that these small RNAs provide us. So again, there's yet another overlap and connection for more combinatorial regulation, and there's actually other connections between this and this that I'm not going to try to give you here. So, okay, so the final sort of example of networks that I'll tell you about is one of the complex and very easy to look at in some ways behaviors in bacteria are things like motility. So this is new work from Nicholas DeLay, and he took the RNA landscape approach, that is the set of plasmids that we have that express these small RNAs, but instead of looking at a gene expression for the moment, he's looking at motility. And what you can see here, so this is really easy, you put it on low density agar, and the bacteria swim out, and if they can't swim, they don't, and you can measure how far they swim, and using that kind of quantitative test, what he found was a whole bunch of small RNAs inhibited motility when they were overexpressed, and some of those clearly affected when it's growing under normal conditions. So now can we take that data and try to start to understand how it's working, and there's the very complex regulation of the genes involved in motility, but at the very top of the cascade of regulators are transcription factors FLHDC, so he made a translational fusion to that, and I've just taken the relevant data here from the last slide. The guys that downregulate motility for the most part regulate this. So we have now multiple, and remember, each one of these, so this is regulated by a different transcriptional regulator. So now we have multiple transcriptional regulators feeding into regulating motility through small RNAs and also frequently through other levels as well. So I think I'll sort of sum up in the next few slides. As we look more and more at these small RNAs, we find that they provide a lot of connections between regulatory cascades. They're regulated by all of our favorite transcriptional regulators, specialized sigma factors, two-component systems, repressors, activators. There's nothing special about the upstream regulators. They just use everything that's there. And they're embedded in these regulons that we've been studying for years and not realizing had small RNAs in them. There are multiple small RNAs can regulate the same target, and each small RNA has multiple targets. So we have a lot to still find out. And the computational challenges, probably finding the small RNAs from genome sequences, maybe that's the part that's the most under control. It started out as an important computational approach to getting the first ones that allowed us to sort of extrapolate to others, but there are still some interesting possibilities there. And it's still a bit of a challenge to sort out the 5' UTRs that look like functional RNAs from the things that work in trans, and sometimes they may do both. Matching small RNAs to target mRNAs is clearly one of the major challenges that we face, and that so I've talked about E. coli. These small RNAs are in every organism. They're not always so easily identified as pairing because we don't always have HFQ as the hook to say that they're pairing RNAs, and there are ones that don't use HFQ that pair. But the first thing to think about in the function of a small RNA is that it would pair with a message, and we'd like to know that. So if we could predict it better, we might be in much better shape. And I think the kinds of approaches that we've heard about finding better structures will actually help us a great deal in doing that, because it's clear with the ones that we know about already that the structural constraints on the messages and on the small RNAs are helping to drive the interactions, and we don't have those in our programs at the moment for predicting structures. And finally, for those of you interested in regulatory networks, we can start to draw the lines between these things, but what the contributions are, we're going to need a lot more numbers on to get these really clean. And the different regulons that we're used to sort of thinking of more independently are clearly crosstalking a great deal through small RNAs. So I will, the work that I talked about, I realized as I was sitting there, I've left a number of people off this slide, but the people in red are the people who did the work that they talked about today. We have a long-standing collaboration with Gigi Stortz's lab to do global searches and to think about small RNAs and small proteins, and I'd be happy to take any questions.